In this session, we are going to discuss about the structure of human egg and then various types of egg in various animals in the next part. Human egg is the structure that is small in size and that is about zero point 0.1 mm. It is round in structure. It has its own plasma membrane. This plasma membrane is known as vitelline membrane. So, either plasma membrane or vitelline membrane we can call it, and it is secreted by the cell itself. On the basis of this, the terms are given that which part secretes which membrane, like primary, secondary, tertiary. So, this first membrane which is formed by the cell, we call it also as a primary membrane. So, primary membrane which is secreted by the cell, also called as vitelline membrane. The cytoplasm is divided into two parts. The outer part is called as egg cortex and it is less dense, and we can also call it as exoplasm, whereas the inner one where most of the organelles are there, the cytoplasm and the nucleus is also there, that inner part is called as ooplasm. So, this is the ooplasm and the outer part is egg cortex. So, an ooplasm has nucleus and all organelles and also has nutritive content, that is yolk, and in case of human it is very less. And nucleus, we call this nucleus as eccentric, that is bulging, and it is not in the center of the egg, it is on the one side. This nucleus is known as germinal vesicle. This nucleus is known as germinal vesicle, and because of the position of the nucleus, we have given the polarity to this. So, the side where the nucleus is there, this side, we call this side as animal pole, and the entry of the sperm into the egg always takes place through this animal pole, and the opposite side would be termed as the vegetal pole. So, egg has polarity, one side where egg is and the other side is the vegetal pole. Outside this vitelline membrane there is a non-cellular layer, and this is secreted by follicular cells, that is why it is called as jelly membrane. So, because there are no cells, this layer is known as zona pellucida and it is secreted by follicular cells. Now, the third layer is actually made up of follicle itself, and they are arranged in such a manner that they appear radiating out from the outside. That is why it is known as corona radiata, and it is made up of follicular cells itself. So, one layer is secreted by this cell itself. So, we see here that the first layer was secreted by the cell, vitelline membrane, secondary membrane secreted by follicular cells, and tertiary membrane is secreted by the glands, or oviduct, that is the example is the shell. So, the third layer which is made up of follicle itself, they are arranged in a this membrane form, and that is what we call here as the corona radiata. So, this cell appear radiating out from the central part, and I have drawn I have not completed this full part, but for the entire part is filled with the corona radiata follicular cells. So, in case of human eggs, the cytoplasm is divided into two zones, the complete organelle and a little bit of yolk, whatever is present, as present in the central part of the cytoplasm. The nucleus is shifted to one side, and this is a haploid cell, this is the female gamete. On the basis of from where the membranes they are formed, we can divide the membranes into three categories: the egg envelopes or the egg membranes. They arise from different structure. So, we have given them the category. So the first category is primary membrane secreted by cell. Example is vitelline membrane, and it is the primary membrane of egg. Secondary membrane is the second category, which is secreted by the follicular cells or made up of these secreted by or made up of follicular cells. So example is zona pellucida and corona radiata. Zona pellucida is secreted by follicular cells, whereas corona radiata is made up of follicular cell itself. And the third category is of tertiary membrane. They are secreted by certain glands, especially in case of the oviduct part, and the example over here is the shell. Now we see many eggs which have shells around them. So shell is secreted by glands. We also call it as shell gland, which is towards the end of the oviduct, that is outer protective layer. And this is how the egg membranes are classified into three categories: primary membrane, secondary membrane and tertiary membrane. In case of human egg, yolk content is very less. So such eggs where the yolk content is very less, we call those eggs as microlithial eggs. So when the yolk is very less, in case of human beings, we call those eggs as microlithial eggs. And as the quantity is very less or negligible, we call micro means very less. In case of other animals, for yolk synthesis, there is a special type of chromosomes which are present, and these chromosomes, they are called as Lamb-Brush chromosomes. They appear and they help in yolk synthesis. It is not seen in case of human beings. Lamb-Brush, they appear only in lower vertebrates, and the role of these chromosomes is to increase the yolk synthesis. So the structure of this human egg or ovum is spherical. There are two poles on the basis of the position of the nucleus. There two names are given, that is animal pole and the vegetal pole, and the sperm enters through this animal pole. Now the significance of the entry of sperm from this animal pole is that the first cytokinesis would be on the same plane. So that means the first zygotic division would also be on the same plane, but entry will be from this animal pole. So as soon as the sperm enters the nucleus, which has half the number of chromosome, would fuse with the chromosome present in the egg or ovum, and fusion can take so it is present here, this nucleus is present here, animal pole. So as soon as possible, there is a, after the entry, this fusion takes place. So that is why the entry takes place from this animal pole. And in the next next part, we are going to talk about different types of eggs in different animals. Thank you.